Ok, me parece que ya estamos en vivo. No sé si a todos les aparezca, pero bueno. Siendo de esta manera, primeramente, esperemos que las personas que nos empiecen a sintonizar estén ya preparadas para lo que se verá el día de hoy. Así que muy buenos días. Y para continuar con el programa de hoy, a nombre del señor embajador de la UNESCO, Jorge Herrera Ramos, chairman del World Congress of Neurosurgery, de la UNESCO Absnett Walter E. Dandy, in honor of Professor Yasargil, a quien agradecemos por la organización de este magno evento, así como a la doctora Deyanira Capi Casillas, chairman del capítulo ANS y PN, al doctor Salim Abdurav, chairman de la Walter E. Dandy Neurosurgical Society, al doctor Babak Kateb, chairman de la Society for Brain Mapping and Therapeutics y de la Brain Mapping Foundation. Todos ellos vice chairmans del comité organizador, y al doctor Parmeides Guadarrama Ortiz, chairman del Centro Especializado de Neurociencias y Neurocirugía en México. Bueno, es para mí un honor presentar la siguiente ponencia. Una segunda oportunidad para la exploración de regeneración y restauración neural, que corre a cargo del doctor Christopher Burtan, desde Rumania. Perteneciente a la Romanian Association of Neuroscience, and a student at the University of Medicine and Pharmacy, Carol Davila of Bucharest, Romania. Also, we're gonna present another topic. Y para esa presentación vamos a tener al doctor Guadalupe A. Martínez Sánchez con interacciones cerebrales en el proceso educacional durante la pandemia del COVID-19. El doctor Guadalupe es cirujano académico de la UNAM, con grados en Medicina y Enfermería, profesor en la Escuela Nacional Preparatoria UNAM, con grado en Psicología, y es organizador y coordinador general del curso de inducción a las ciencias médicas y de la salud.
Good afternoon or morning, depending on where you're listening from. I'm Dr. Sebastian Dubrescu, and it is my pleasure to open today's presentations. Before we start, I want to welcome you to the third week of the World Congress of Neurosurgery, UNESCO Walter E. Dandy Society, in honor of Professor Ghazi Yasargil. On behalf of the ambassador of UNESCO ASP.NET, Jorge Herrera Ramos, President of the World Congress, whom we thank for organizing, the Vice President, Dr. Deyanira Kapi, President of the Untitled Chapter, Dr. Salim Abdul Rauf, President of Walter E. Dandy Neurosurgical Society, with Dr. Babak Kateb, President of the Society for Brain Mapping Therapeutics, Brain Mapping Foundation, Honorary Chairman of the Committee, and Professor Nasser Agandur, Professor Marcus Rota, and to Dr. Parmenides Guadarrama Ortiz, Director of the Specialized Center of Neurosurgery and Neurosciences of Mexico. It is an honor to present today's mediator, Dr. Cristina Sari.
Thank you, Dr. Sebastian Dobrescu. It is a pleasure for me to be here today again with you. And it's an honor to present today's presentation, Dr. Christopher Burtan, member of the Romanian Association of Neuroscience and student at Carol Davila University of Medicine and Pharmacy of Bucharest, Romania. With the topic, A Second Chance for Neuron Degeneration and Restoration, welcome Dr. Christopher Burtan.
Thank you very much, Dr. Cristina Sari, and hello everyone. Thank you for the kind introduction. I would like to also thank all the coordinators who have organized this Congress, especially Dr. Jorge Herrera Ramos and Dr. Salim Adrarouf and Dr. Babak Kateb. I would like to start by presenting the screen. Are you seeing this? Are you seeing the screen very well?
Yes, Dr. Burtan.
Thank you very much. Okay, so good day, everyone, and wherever you may be. I would like to start with to explore, the possibilities of neuroregeneration and restoration within the world, and how it may help millions of people across the world suffering from neural degeneration or even spinal cord injuries. To the right of the screen, you see a brain mapping image. And actually, this is very indicative of the complexity of the brain. And this doesn't even show everything. And you can see the connections between various regions of the brain and the hurdles and the difficulties that neuroregeneration, at least within the brain, has to overcome. And this is something that we will discuss later. I'd like to start off with a few words about regeneration that we see in nature, specifically the regeneration we can model and learn from. One such creature is the planarian, which is a type of flatworm. A research group in Germany actually demonstrated a deep intrinsic characteristic of the planarian regeneration facilitated by the abundant population of adult stem cells, and can further our understandings of the molecular underpinnings such as WNT and slit axis signals, which the human body shares. So this animal model can be used as such. What's fascinating here is that the survival and the growth of the planarian from amputations, you can see the cross section here in various different sizes and shapes, it all ended in a partial or complete regeneration. You can see these two black eyes, or ocelli as it's called, at one end of the regeneration mechanism. Moving on to another animal that vies for the title of master of regeneration, we have the axolotl. It's quite a cute little character here. And this amphibian is actually a marvelous and complex example of the potential mechanisms of neurodegeneration and regeneration, specifically. As we can see, after an initial amputation, the limb successfully grew back. You can see, it growing. And this happened amazingly over 31 days. Just imagine, growing a limb in 31 days. I would love to be able to do such a thing, but unfortunately, some systems rein that in. The regeneration of the peripheral nervous system is actually a much easier key distinction here. The growing of the limb is that we'll discuss much later. So, neuroregeneration, what is it? It's the term refers to the ability of the neurosystem to alter, renew, or replace itself after injury or disease. This ability is in the central nervous system, but more prevalent in the peripheral nervous system. And we'll see differences between the central nervous system and the peripheral nervous system. Here, there are some outlines of areas that need to be considered and the techniques and processes that are required for a successful treatment of neuronal disorders. The difference between tissue repair and regeneration is the migration or the growth path of these cells. Tissue repair usually stays within the confines of the tissue type, while for specifically neuronal regeneration is a complex multi-layered process involving degeneration, clearance, and environmental preparation. The methods that the tissue and cells rely on can be both extrinsic and intrinsic. So the cells that release molecules and the cells that respond to other cell types, such as support cells within the central and peripheral nervous system. Since I've mentioned that there's a very big difference between these two, the cellular makeup and actually the molecules that are expressed within the two are very different, and these are actually among the difficulties within these two systems. Alongside this is the immune response that plays a key factor in any injury that a person might have, for example, spinal cord injuries. And so you can have inhibitory role or facilitating the growth of neurons. For example, serotonin and histamine are secreted by the endoneurium, and within the peripheral nervous system, this actually leads to the recruitment of macrophages. And we'll discuss this a bit later as well. Transplantation is something well known to the surgery community, that you graft neurons to each other to help neurons that are from one side to another, usually very big cuts or injuries that are not easily pieced back together. And finally, we have the effects of neuroplasticity, so the growth of, say, dendritic spines, which actually allow for the new connections to be formed. And actually, we'll see this in neurogenesis as well. And epigenetics plays a very key part, and something we'll talk about when we talk about stem cells and the way we tackle that. The process of repair within the nervous system can be very complex. There are various mechanisms that promote and guide the growth of neurons. Neuronal repair can be achieved in a number of ways. In the image on the right, we can see the development of neurons in a dendrite gyrus of 7 to 10-week-old mice. These newly generated granule neurons were induced to grow through a retrovirus-mediated gene transduction with CAG GFP. Neurons labeled with green fluorescent, so you can see here the green all over, it was used to visualize them. So the samples were taken at various days post-infections. So you can see here, this is at 3 days, 7 days, 10, 14, 21, 28, 56, and 14 months. And the red stain is actually, it's a DAPI stain. This DAPI stain is just to visualize live or fixed cells. And so it's a contrast agent that allows you to see the different layers within the dendrite gyrus. If you look at A and B, you can begin to see the axon process. So if you can look at where my pointer is, you can begin to see the processes extending out. And it's not a definite movement. There's no exact location. They move up. It's all over the place. But you can see where they're going eventually. In B, you can generally see they're moving through this granule layer. So at 10 days, you can begin to see actually they reach the CA3 region of the dendrite gyrus, or it's also called the molecular layer. You can see the axons finally reaching this layer much later on, and this is important in the sense that potential connections can be connected to the spines. So at 14 days, it was discovered that between 14 and 15 days, dendritic spines could be seen within these arborizations. And it peaked about a few days later, so around 21 days, which does actually provide some evidence of synaptic input and some neuroplasticity. So this might be an opportunity for people to regrow neurons that are deficient in certain neurodegeneration or neuro conditions. The dendritic and the axonal arborization was just as extensive, you can see here, like in normal granule neurons. However, they only reached the initial stages of neuronal maturation, expressed only immature neuronal markers. From this, we can take away that the neurons can be induced to grow, but the functional connections they make can be a bit uncontrollable, or it's very hard to know which neurons they'll connect to or synapse with. From this, we can take away that neurons can be induced, the ability, for the adult neurogenesis is a bit contested, because there's some studies which find evidence, especially in mouse studies, and there's other evidence in, for example, human and monkey studies, which hasn't found any evidence of neurogenesis in brain samples. Moving on, I want to talk about quickly about why neuroregeneration is very important. So every year around the world, there's about 250,000 to 500,000 people that suffer a spinal cord injury. The causes of spinal cord of injury to the spine can be traumatic. In particular, motor vehicle accidents, including as well as slips and falls. The other is actually non-traumatic, and so these include degeneration and infections. The location of the spinal cord injury is very important to the prognosis of the patient, with cervical lesions most likely causing quadriplegia. Lesions at lower parts of the spine affect their respective sensory or motor terminal places they supply. The spinal cord injury is classified under two categories, complete or incomplete. A patient in complete spinal cord injury has no motor, no sensory function with no sacral sparring. The patient with incomplete spinal cord injury has varying levels of sensory or motor functions, with a few subtypes that indicate what the prognosis will be. For example, anterior cord, central cord, and Brown Sequard syndrome are the most common types of incomplete spinal cord injury. As an example, one side of the spinal cord is affected in the Brown Sequard syndrome, which means a loss of ipsilateral motor function and contralateral loss of pain and sensation. Ipsilateral is the same side, contralateral is the opposite side. The prognosis of spinal cord injury is usually quite good. An investigation must take place in a clinical setting to elucidate the level of muscle power that the affected person can exhibit. In this circumstance, delivering some sort of regeneration within a short time would actually be very, very useful. Imagine that there's a big trauma, and you'd be able to sort of immediately be able to try and repair it, or at least mitigate the damages. And some of the damages can be quite debilitating. I would like to talk about later about the restoration of upper limb movement as well. So this is one of the clinical applications, in that you can actually restore clinical upper limb movement via an artificial connection. And this is actually a fairly recent technology that is coming to the fore. As you've heard from my colleague, Dr. Michael Basil, amyotrophic lateral sclerosis, or motor neuron disease, is characterized by a progressive and irreversible degeneration of motor neurons in the brainstem, cerebral cortex, and the spinal cord, leading to muscle weakness, dysfunction, and loss of mobility. Its prevalence is approximately 1 in 50,000, and a recent study in Canada has found actually the incidence is increasing by 8% in adults and 10% in children over a 11-year period. ALS is actually currently untreatable. However, ALS has been the focus of neural stem cell strategies in two main ways. One is to try and rescue endogenous motor neurons from the disease-related degeneration, and the other is for the neural stem cell transplantation to survive and integrate into the host nervous system. Parkinson's disease is a neurodegenerative disease that leads to progressive degeneration of dopaminergic neurons in the substantia nigra of the midbrain. The current treatment, as such, involves the L3,4-dihydroxyphenylalanine, or as it's more commonly known, L-DOPA. But its inability to cross the blood-brain barrier leads to the resurgence of Parkinson's syndrome. Long-term relief, either by neuroprotection of dying dopaminergic neurons or replacement of dopaminergic neurons, is hoped to be achieved by stem cell transplantation. Studies into stem cells and Parkinson's differ in the effectiveness to innovate and restore functionality to the striatum, leading to increased dopamine release. And here we also have age-related macular degeneration, which follows a same basic principle as such. And as of now, neurosurgery may help control the severity of some of these neurodegenerative diseases. We can move on to how the human body actually responds to regeneration, specifically the difference between the peripheral nervous system and the central nervous system. And there is a big difference, and the environment between the two that facilitate or inhibit the growth of axons. Keep in mind these differences. They are actually very vital to overcome the guided or artificial neural regeneration. On the left here, you can see the schematic of the peripheral nervous system. So you can see here there's a cut, and everything distal from that cut withers away or degenerates away in a process called Wallerian Degeneration. The presence of Schwann cells here, these are the myelinating cells of the axons, actually can guide the growth of axons to their end organ. On the right here, you can see the supporting cells for the central nervous system, so astrocytes here in blue, and also microglia here in yellow. They also produce oligodendrocytes, sorry, they produce the myelin sheath, whereas the astrocytes and microglia, they actually secrete inhibitory signals to actually disrupt this growth. And this is actually a neuroprotective role within the central nervous system. So something I want you to keep in mind is actually this is the environment that is the limiting factor in the ability for the neurons to be repaired in the lesion and the injury. So the regenerative capability of the peripheral nervous system is actually fairly well known. And I would like to start with an overview of the regeneration process and the reason why regeneration of peripheral nerves are relatively easier. These are hopefully some of the considerations we can hopefully transfer over to central nervous system nerves, which are long thought to be unrepairable. In this case, we actually begin with a cut. Here you can see in A, you can see the cut. It can also be a crush injury, which is actually much easier to regenerate. There is also a protective mechanism of the cell body. For example, if you just imagine there's a cell body, the nucleus will actually move away, and the ER, the endoplasmic reticulum, and the missile bodies will actually undergo chromatolysis and cause edema of the soma. This will actually help preserve the neuron until a functional innervation has been established. We can move on. So from B, C, and D, we can see actually Wallerian degeneration of the nerve that begins soon after the cut. And we'll talk about that more in the next slide. So if we have an injury that is more distal to the soma, the cell body, it's a very good chance actually that the entire neuron will survive. It will actually continue. Whereas if the injury is actually very, very close to the cell body, then it may cause the cell body and the entire nerve to die. The Schwann cells themselves, you can see they're in blue, these blue little dots surrounding within the endoneurium, have a remarkable ability to change their phenotype in response to trauma, and along with recruited galactin-3 macrophages, clear the debris in preparation for the axon growth cone. So you can see the growth cone in beginning, this bud here, and then the growth cones, the growing axon projections away from the node of Ranvier. The axon sprout then can begin, and the Schwann cell actually form a band across the endoneurium to actually guide the path of one of these axons, and it will eventually reach its destination. Whereas most axonal sprouts will not connect with their previous sensory or effector organs, and so will degenerate away. Complete failure of a creation of functional connection is actually called a neuroma, and it's seen more in more traumatic injuries, such that the endoneurium is ruptured, and so the axon will actually start growing outside of the endoneurium and form no connections. The rupture of the endoneurium actually develops into a glial scar, and we'll talk about glial scars in a moment. And this is why grafts are actually very important. They actually connect one end of a neuron to another to actually guide and help facilitate this axonal growth. And eventually, right at the end in J, you'll see that the functional connection is made and the nerve is completely remyelinated. So the first step in the degeneration is Wallerian degeneration. It's a degeneration of the axon distal to the site of injury. So if you see here, these bright white dots, beyond that is the distal axon. It's a biphasal process with a latency phase of around 34 hours. So you can see here, after a cut, there has been, you can see no visible degeneration. Then at a certain signal, which is considered to be the cessation of survival factors, the fragmentation phase begins. And you can see very slightly that there begins fragmenting and fragmenting and fragmenting. So this all happens within a period of about 40 minutes, but the time is variable. And I should mention that this is actually a mouse neuron, which is much easier to model. The precise molecular pathway of Wallerian degeneration is not yet fully known. However, in some studies some studies with genes that code for slow Wallerian degeneration can actually extend the latency period by a few days. And a similar degenerative process occurs in the central nervous system and can contribute to poor regenerative the environment of the central nervous system. And this is actually a recurring theme. Remember, the environment is of these lesions are a big difficulty problem. Another difference within the peripheral nervous system I would like to highlight is the ability, well, firstly, it's the actual presence of Schwann cells. They are in the peripheral, and then you have oligodendrocytes in the central nervous system. The ability of Schwann cells to change phenotype and in response to chemical signals is very, very important. In this case, I'm showing the effects of cyclic AMP. But the transcription factor c-Jun is the driving force in reprogramming denervated Schwann cells to upregulate genes implicated in promoting axon growth. Axon growth, neural survival, and macrophage invasion. So all those three, they do all one together just to promote that Wallerian degeneration I talked about earlier. They also activate mechanisms to break down their myelin sheaths and transform morphologically into cells with long parallel processes. So imagine this is a control cell. So this is a myelinating cell. Just imagine long processes away from it. It's not actually seen here, but this is actually a good representation of the vast changes in phenotype. The increased expression of a myelinated associated genes has been linked with high cyclic AMP levels. In the case of regenerating neurons, cyclic AMP levels would actually decrease. So in the image on the top right, we can see a dramatic difference between the control, so the normal, and after Schwann cells have been exposed to a variant of cyclic AMP. So the first two are actually early phase Schwann cells. The next two are middle phase, and these last four are late phase. And these are markers, these CNPAs, sulfate antigen, galactocerebroside antigen, and all of these antigens, or these markers, indicate the level of differentiation. Returning back to actually CGen, and I just want to remind you about this trophic factor is very important to actually reverse the differentiation. So actually, these Schwann cells are actually differentiated cells. They produce myelin, and so CGen will actually reverse the differentiation. So you can imagine just a sort of reverse of this, and will downregulate myelin genes and upregulate markers of immature Schwann cells. The lower image is done by transmission electron microscopy, and it illustrates the different phenotype of a normal Schwann cell, which is this one, and then the one that was expressed, shown with cyclic AMP. Schwann cells can actually also clear the debris that actually the neuron degeneration and the myelin degeneration cause. So the fragments are all cleared up by the Schwann cells. So along with the macrophages, it can help in a wide set of roles. It can also control the generation process of the axon via the expression of CGen, which determines the expression of trophic factors, adhesion molecules, and the regeneration tract. So in comparison to the oligodendrocytes, in a genome-wide expression analysis study that increased the expression of myelin-associated genes, contributed to the pathophysiology of schizophrenia. Okay. As we discussed, axon regenerated, remyelinated, and eventually innervate target organs. How complete this process is, and the extent to which the organs are innervated by the correct axons, is related to the degree of functional recovery. So after phagocytosis of myelin and axonal fragments, the Schwann cells align into bands of Bungner, which guides the regenerating axons back to their synaptic targets. In image A, we can see the pictomicrograph of YFP labeled axons regenerating in the tibial nerve following a sciatic nerve crush. The growth cones are indicated via the white arrows. An electron micrograph of the transverse sciatic nerve 10 days later, so in B, shows axons with a diameter are greater than 1 micrometer, where Schwann cells have begun the process of remyelination. Please note that the myelin sheath at this stage is much thinner than the uninjured state. So they're still growing, and it eventually get almost to normal. The white arrow here is actually myelin debris that's still being degraded by macrophages. C is actually just a overview of a schematic of how exactly the growth of the axon develops. So it shows that the directional nature of the communication between the growing axon and the Schwann cells. Schwann cells may not only provide the means to metabolically support axonal growth, but also modify the axons themselves. For example, axonal absence in the case of severely injured people affects the expression of growth factor, the growth factor glial cell line derived neurotrophic factor. This is clinically important since the speed of axon regeneration slows down, leading to poor functional outcomes. So we can move on to the difficulties within the regeneration strategies. So I've outlined the Schwann cells, some of the inhibitory molecules, and the glial scars, as you can see here. So within the CNS, actually, axon regeneration fails entirely. So this is this is part of the oligodendrocyte issue. It fails for two main reasons. Firstly, the environment surrounding the lesion is inhibitory to axon growth, and the second is that regeneration potential of the CNS neurons are severely reduced. Development of secondary degeneration, which is caused by imbalance of amino acids, ions, reactive oxygen species, and inflammatory markers in the immediate area of the injury, causes further damage that can lead to formation of a glial scar. So the glial scar is actually formed from a cocktail, or it is initiated from a cocktail of growth factors, interleukins, and some other such trophic factors, such as transforming growth factor alpha, ciliary neurotrophic factor, and interleukin 6, among others. The glial scar is both beneficial and detrimental. The astrocytic produced proteoglycans in the glial scar inhibit the conduction of action potentials in undamaged nerve fibers close to the spinal cord injuries, as well as preventing axonal regeneration in these areas. So you can see in the image to the right, we can see a glial scar 9 months after spinal cord stab lesion. So you can see what the aftermath of a stab here. So in image A, we can see a sagittal section of the spinal cord, illustrating the astrocyte hypertrophy in red, so all this, and a molecule called chondroitin sulfate proteoglycan upregulation. So you can see in blue. Actually, you can see it all over, but it's sort of masked by this red astrocyte hypertrophy. In B and C, you can actually see that B and C is more magnified sections of A. You can see the extreme hypertrophy of astrocytes, and also the presence, a better view of the presence of chondroitin sulfate proteoglycan still remaining. So you can see here the blue is sort of forms this purple color when it interferes with red. In D, the micro-transplanted dorsal root ganglion neurons can actually grow almost everywhere within the growth zone of Wallerian degeneration, and can react, and within the reactive gliosis. So gliosis is more glia. It just means that the more glia is being formed, and this is actually astrocytic gliosis. And you can see here that the neurons actually form a rostral to the lesion. However, the regrowing axons, and you can probably see some regrowing axons here in green, are excluded from this very extremely dense fibrous reactive astrocytes. So everything to the right of this area is a very dense connective tissue. And within this, at this point, that there's an absence of proteoglycans. So just remember, this is actually a transplanted dorsal root ganglion above this lesion. I mentioned the beneficial effects, and actually the reason why glial scars are very, very good is that it serves to repair the blood-brain barrier to prevent an overwhelming inflammatory response. So this separation between healthy and injury prevents uncontrollable tissue damage. I would just like to show that in a micro-transplanted neuron, this image shows the failure of regeneration of micro-transplanted dorsal root ganglion axons on reaching a proteoglycan gradient. So we talked about the chondroitin sulfate proteoglycan, and you can see that there's a big concentration gradient. So all this in blue, and which actually stops the axon from growing. So the transplanted adult sensory neurons were capable of extending axons rapidly at about 1 millimeter per day. So among the oligodendrocytes, degenerating myelin, and the astrocytes that were intensely reactive, owing to the accompanying Wallerian degeneration. So it follows the path of the previous axon. However, it is important to reiterate that the site of transplantation must be far enough from the lesion to be beyond the zone of serum leakage through the ruptured BBB, so the blood-brain barrier. This double scanning confocal pictomicrograph of a transplanted adult dorsal root ganglion, so the neurons are in green, extend into the periphery. So here you can see the inner spinal cord, and here's the periphery, because of the presence of chondroitin sulfate proteoglycan deposition. So you can see large amounts within the dorsal root ganglion. They are actually unable to transverse through these regions. Reactive astroglia, actually, that are distal to the lesion, so just imagine lower than this lesion, are surprisingly growth supportive, and the capacity for regeneration can last for many months until glial hypertrophy comes into play. So I've painted a small picture about of the environment that leads to the difficulties in regeneration in the central nervous system. A number of studies are looking into creating a gel matrix-like medium to facilitate the growth of nerves in larger trauma sites within the spinal cord. And again, this is to do with the environment. You want to do the correct proliferation markers and or disable any inhibitory markers. The inhibitory molecules within the matrix of the spinal cord are actually secreted by the macrophages, microglia, and the astrocytes. The term peripheral nerve conditioning lesions signifies the upregulation of regeneration-associated genes and the production of structural proteins for axonal outgrowth. The conditioning lesion sets up an environment highly conducive to regeneration. On the image to the left, from a recent study of axonal regeneration into neural progenitor cell grafts, conditioning lesions activate intrinsic neuronal growth state. So in A and B, you can see that there's an uptick in conditioning, peripheral conditioning lesion. So the GAP43, which is actually a molecule that basically grows the axon growth cone, and it shows you that within this ipsilateral dorsal root ganglia, it is growing. You can also see CGen here, so it's upregulated, and as I said, it relates to the expression and expression within Schwann cells, and they help grow the axons. And so within this injury, the ipsilateral side is growing, but it can't contralaterally. And so the growth-associated protein 43, that's the GAP43, it's expressed in neuronal growth cones, and it is involved in the plasticity of the neurons. And so within the peripheral nervous system, these regeneration-associated genes are upregulated. The molecular regulation, the molecular regulation of the neuronal growth is inhibited mainly by myelin-associated inhibitors. This family of proteins includes no-go proteins, and one of them is no-go A. You have myelin-associated glycoproteins and also oligodendrocyte myelin glycoproteins. And all of these inhibit axonal growth. The genetic deletion of no-go A actually promotes cortical spinal and raphe spinal tract growth and enhances the functional recovery after spinal cord injury. Myelin-associated glycoprotein interacts with several neuronal receptors to limit the neurite growth, outgrowth in vitro. The neuronal type is very important in determining the inhibition of neuronal growth. For example, postnatal cerebellar granule neurons, ganglioside GT1B receptor appears to be more important. These inhibitory molecules secreted by oligodendrocytes activate the protein kinase C pathway, and you can't see here, but it also works via Rho, and it leads to the inhibition of cytoskeleton development and the growth cone collapse. Dibutyryl cyclic AMP, so you can see here CAMP here, actually inhibits the action of these molecules. You can see effects on the inhibits the growth cone collapse, and acts on the cytoskeleton development within the axon, so it allows it to grow. And finally here, chondroitin sulfate proteoglycan can create a gradient within the glial scar region for many, many months after injury, which inhibits the extension of axon processes into these regions and affect the conduction of action potentials. Reactive gliosis is included in the conditions within lesions inhibiting axonal regeneration. So there are a number of strategies to tackle the problems mentioned previously. In current surgeries involving motor and sensory repair, grafting is the main process to regrow or help grow the axons. Within the last few decades, the interest into stem cell research and its clinical use has increased year by year, and its delivery and activation is important in a successful connection between the peripheral nervous system and the central nervous system. The lentivirus here, lentivirus system, as shown in a simplistic diagram, is a tool for gene delivery and gene therapy. This is a virus that is derived from the Retroviridae family of viruses. From the schematic here, you can see the process that delivers the viral cDNA that replicates the inputted sgRNA constructs into specific targets. So you can actually provide or make sure that it goes to the target you wish. This process allows for these viruses to reverse transcribe into the host DNA genome. The lentivirus contains 3 main genes. You have GAG for the viral assembly and infection. You have POL, and like polymerase, it encodes for reverse transcriptase and integrase, so it allows to replicate and integrate into the cell DNA. And the ENV gene, that determines tropism and enables entry into the cell. There are also 2 regulatory genes. You have TAT and REV. The lentivirus system can be tailored to the requirements of the study as long as the gene of interest, the promoter, the tag, and or the reporter is supplied. So as long as you have these 4, you can actually target a specific cell you wish to. Transplants of neuronal restricted precursors together with glial restricted precursors have been examined for for the capacity to form a functional relay between injured dorsal column sensory axons and their targets in the dorsal column nuclei in the brainstem. A lentivirus expressing BDNF was injected into the dorsal column nuclei to generate a trophic gradient for directional axon growth. Parkinson's disease, which is characterized by CAG repeats in the dopaminergic neurons, can actually be targeted by the lentiviral system, and it can offer a way to limit the severity of the disease. Here, I just want to quickly present a graph of the sural nerves. You can see here the sural nerve graphs to connect the two radial nerve stumps. The patients with radial nerve injury presents with paralysis of the wrist, the thumb, and the finger extension. From this image, I would like to just present the idea that the growth of the axon of the radial nerve uses the sural nerve graft as a guide to grow. On the left, you have an intraoperative view of the dissection of the medial aspect of the upper arm, showing neural grafting of the radial nerve. After medial transposition of a short distal stump of the radial nerve, a third incision over the distal part of the biceps was needed to complete nerve coaptation. A nerve coaptation is basically the connection between two nerves, to align the nerves together. On the right, you see an intraoperative view of the dissection on the medial aspect of the upper arm, showing sural grafting of the radial nerve. So another one. The distal stump of the radial nerve was medially transposed, so it's moved. In the context of regeneration, the sural nerve acts as a guide for regenerating axons to meet back up with the endoneurium of the distal radial nerve. In some patients in whom the proximal stump of the radial nerve is inaccessible, alternatives such as a nerve transfers are a viable alternative to restore triceps functions. Just to mention here about a very important medical field that is gaining traction as new techniques and research are explored, which is called stem cell research. They are seen as a next step into targeting differentiation and maturation of stem cell-derived neuronal lineage targets. The ability of self-renewal and the differentiation potential are two properties of stem cells that give the ability to develop into all other cell types. There are three main locations or types of obtaining stem cells. The first of which is the adult stem cells, which are located in the hippocampus, the subventricular zone, and others are a bit more experimental. In the past, stem cells were widely harvested from embryos, but the advent of induced progenitor stem cells has rendered this method as a greater ethical concern. The opportunities for regeneration, and not just neurons, but other tissue types, are actually massive, and research into controlling the differentiation of correct cell types are still ongoing. As you can see from the images, stem cells can differentiate into progenitor cells. So here you have a human-induced pluripotential stem cell, which has a particular morphology after it differentiates once, and then it finally differentiates into neuroprogenitors. In the lower image, after 1 week in the culture, many of the mouse stem cell derived neural stem cell derived cells exhibited pyramidal morphology and extended processes. The regulation of neurons within the lesions and regenerating axonal buds are controlled via opposing signals of proliferation factors and degenerating factors. So here we have two very important ones. We have fibroblast growth factor 2 and epidermal growth factor, and these both induce stem cell proliferation. We also have found that other neurotrophic factors which are specific for various regulation. For example, brain-derived neurotrophic factor is very important in myelinogenesis regulation. There's also a ciliary neurotrophic factor, which actually helps in motor neuroprotection. Also, neurotropin 4 as a small example for sensory neuroprotection. The glial cell line neurotrophic factors can actually counteract certain conditions and actually help in neuron survival. For example, in ALS, so the prognosis for ALS is usually about 2 to 4 years, but with stem cells, you can actually probably delay the prognosis. It is possible to reprogram somatic cells via molecular signals through transcription factors that control differentiation into induced pluripotent stem cells. These cells can then further differentiate into neural progenitor cells. So the image on the right shows the expression of TUB3 in induced pluripotent stem cell derived neurons differentiated from neural stem cells after 21 days of culture. These neural stem cells can differentiate into 3 neural subtypes: neurons, oligodendrocytes, and astrocytes, so everything in the CNS. In the study, the differentiation from urine-derived induced pluripotent stem cells was carried out by blocking TGF-beta and BMP-dependent SMAD signaling via an episomal method, which is a non-viral vector to express certain molecules. In row A, we can see the morphology of the neurons with their soma and projections and the dendritic and axiomatic projections. The control correctly not having any cells, DAPI is just used to again, as I say, to stain the live cell, so you can see that it actually is present. In row B, we can see a merged image with the green TUB3 immunoreactivity, and you have the DAPI fluorescent stain that shows how the neurons grow on the plate. So TUB3, which is tubulin beta 3 class 3, it's a neuronal marker that almost exclusively is expressed in neurons. It is part of the tubulin family that form and organize microtubules, and therefore have a role in the growth and shrinkage of dendrites and axons. And so you see the growth of axon growth cones within this region. At the same time, OCT4 was confirmed to be present in the same study. The main idea here is that you can harvest specific cells, and we will mention some somatic-derived neuronal stem cells in the next slide. You can harvest specific cells from a patient and grow their own cells. So, I mean, I think that's wonderful. Imagine not needing to wait for a donor or producing your own neurons without fear of immune rejection. From the slide, I would like you to get an idea that induced pluripotent stem cells that derive from the somatic cells. In vitro differentiation protocols were developed for the differentiation of embryonic stem cell cultures into a broad range of neuronal or non-neuronal fates. As you can see here, differentiation into various types. You can get all your somatic cells from fibroblasts, astrocytes, liver cells, can differentiate into many different types. The table on the right shows the various induced stem cells that derive from various somatic cells. For example, the embryonic here, the bone marrow derived, and the adipose stem cells provide a mechanism for myelination greater than the other derived stem cells, while the majority of these derived stem cells prove or provide neurotrophic factors. The problem with stem cells specifically is the conversion efficiency is consistently quite low. So it's less than 0.1%. So you imagine how many cells you're using to get 0.01%. And isolation of individual clones to combat low conversion can display a significant variability in basic parameters such as subtype differentiation, properties, even within a single culture dish. The source of variance of this strange variance actually remains unclear. The induced pluripotent stem cell approach generates a lot of excitement for personalized medicine. So cells derived from the induced pluripotent stem cells could be potentially developed as a autologous replacement cell therapies for various disorders. A wide range of induced pluripotent stem cells, stem cell neuronal types can be developed, including spinal motor neurons, dopaminergic neurons, cortical glutamatergic neurons, GABAergic neurons, and so as to model various neurological disorders. So it's actually quite good for modeling these diseases and helping to understand it. Researchers can actually also get a unique source of motor neurons from individual ALS patients to try and understand how and why neurons die in ALS. Two types of motor neurons are affected in ALS, are upper corticospinal motor neurons and when damaged, they cause some sort of plasticity. Here, I just want to talk about how the lentivirus system was used to express these three molecules into mice. And so you have these mice were converting fibroblasts into neuronal cells. However, when they were put into human cells, they led to cell death. That there wasn't anything they could do to that. However, when they added basic loop helix transcription factor NeuroD1, they detected, they reported spontaneous and evoked postsynaptic currents after about 5 to 6 weeks. So these reprogramming factors actually help with making functional neuronal cells. These, well, let's just talk about how to do some nerve repair. We have grafts and we have sort of bioscaffolds, and these are external mechanisms to assist with the growth of neuron growth. So the graft serves as a bridge for reconstructive nerve to grow across. So you can have allografts, autografts, and neuropathy is just the surgical connection of two neurons together. Within endogenous tissue regeneration is more preferable, since you can have your own cells and you can avoid any potential immune rejection. I mean, regeneration itself takes a very long time. Just imagine waiting years just for a nerve from your spine all the way to your fingers. It's it just takes a very long time. And so most studies to date have focused on the use of biomaterials such as hydrogels to improve the delivery of the cells to the brain. Here I would like to just talk about three alternative research for degeneration. You have here that this was actually a 3D printed sciatic nerve. It was constructed using 3D scanning techniques and reconstructed to actually model an accurate nerve pathway for regeneration, and to bridge the gap between one lesion site to the other. There are a few neurocompatible materials that could be used, including polycaprolactone and silicone. And so you can see here in this lower image that silicone was used to try and bridge neurons together. These actually 3D printed pathways can actually be impregnated with certain factors to to make sure that certain motor neurons or sensory neurons grow in a certain way. Another study found that heat shock protein 27 was actually increased the speed of the regenerative ability of neurons. Talking about scaffolding, scaffold 3 bio 3D circuit, we have the a biological and organic material component bridging two nerves together, and after 8 weeks, you can see the regenerated nerve, and actually not the conduit hasn't fully degenerated into the nerve, but you can see it's far greater than the one that was surrounded by the silicone 3D material, or the material, the biomaterial. After, I just want to finish up a few slides with neural restoration. So what is it? It's actually the external, it's at the, it's an activity to actually restore function of neurons to the body. So you can do this externally, or you can do this, you can do activities. And the current strategies are to rehabilitate people or find artificial devices. So finally, I just want to just go over quickly the few types. You have the rehabilitation, which there are different types. You can do low intensity versus high intensity, compensatory or restoratory. For example, this refers to how you do the treatment. So you compensate, or do you try and completely restore? And you do this via activity, or you do this via computer interfaces, and you can also do this biologically. There's also some neural prostheses that can be helped, and actually you can see that between a rat brain, you can see when it was running, actually produced more neurons. Finally, I just want to just mention that for the final 2 slides, I just want to mention that there were studies into monkeys where they actually could, in a spinal cord injury, they could actually connect the sensory and motor neurons of the wrist all the way to the brain via an artificial interface. And you can see that this is the monkey contracting, and in relation to the AMSC response. The final slide is actually just something that's actually quite interesting, but also this has actually been completed within the last year. This was a study done over 5 years. And you can see that via duplexing signals from the brain, you can actually send brain signals from the brain all the way to the arm. So you can see here you have this red band and bands around the forearm. You can actually restore the sense of touch using these using this machine, and actually decodes, gives you some sensory feedback. Thank you very much. This is the end of my presentation.
Thank you, Dr. Christopher Burtan. May I ask you one question?
Yes, please.
What are the main issues involving stem cells research?
Oh, so in stem cell research, well, that's I mean, it's very in its infancy currently, and so a lot of the problems is actually knowing how to differentiate various cell types into the neuronal lineage you would like to try and achieve. So, for example, even the neurotrophic factors, the exact molecular pathways, they still need to be elucidated. And also, the spine is a lot easier to access with stem cells than the brain, for example. It's actually very hard to be poking into the brain and injecting certain stem cells.
Thank you so much, Dr. Christopher Burtan. I want to thank you once again for this amazing presentation, and I want to thank all the people that were involved in today's presentation. I hope you had a nice time with us, and I hope you have a good day.
Thank you very much, Dr. Christina. And again, I would like to thank everyone for organizing this Congress.